Hey guys, welcome to another episode of the Human AI podcast. Today we're going to talk about blood transfusion and about some pharmacokinetics. So, specifically, some stuff about distribution of the drug. Having said that, let's start a bit with blood transfusion. I'm here with Grog, and yeah, you know how it goes. Grog, are you ready to start?
Yo, what's up, listeners? We're diving into the Human AI podcast, and I'm ready to roll. Blood transfusions and pharmacokinetics. Let's get into it. Want to start with the basics of blood transfusions, like how they work or why they're critical? Or
Yes,
you feeling
both of those questions actually. And remember, Krog, let's keep this podcast nice and brief, three sentences back and forth.
got it, keeping it tight? Blood transfusions replace lost blood, delivering oxygen and nutrients via donor blood. Why are they critical, you ask? They save lives in surgeries, trauma, or conditions like anemia. Your turn. What's the deal with drug distribution and pharmacokinetics?
Hmm. I ask the questions, you say the answers. Um, I'm just a student here. Okay, so when did this start? When did you have the first attempt for blood transfusion?
My bad. I'll stick to answering. The first recorded blood transfusion attempt was around 1628, after William Harvey described blood circulation. It was a messy try, often using animal blood, and results were usually fatal. Anything specific about transfusions you want to dig into
Tell us
next?
more about this William Harvey dude, and what about the Pope in the 1500?
William Harvey was an English physician who, in 1628, mapped out how blood circulates in the body, laying the groundwork for understanding transfusions. In the 1490s, Pope Innocent VIII supposedly got a transfusion attempt with boy's blood to save his life. But it was more myth than medicine and didn't work. Want to explore early transfusion failures or something else?
So how did William Harvey try it?
William Harvey didn't perform transfusions himself. He theorized blood circulation in 1628, proving blood flows in a closed system. His work inspired others to attempt transfusions, often crudely, like using animal to human blood,
How
with
did
pipes or
how
quick.
did he prove it?
Harvey proved blood circulation by dissecting animals and humans, observing heart and vessel functions. He used experiments, like tying off veins, to show blood flows directionally in a closed loop. Want more on his
Yeah,
methods
but
or
how does
some?
it show that it's in a closed loop?
Harvey showed the closed loop by tying off vessels and watching blood flow stop or redirect, proving it circulates continuously through the heart and body. He also measured blood volume pumped by the heart, confirming it couldn't be endlessly new blood. What's next on transfusions? Or are you switching to
No,
pharmacokinetics?
we're gonna stop. We're gonna continue in transfusion for a while. Um, so tell us about Karl Landsteiner.
Karl Landsteiner, an Austrian scientist, discovered blood groups A, B, AB, and O in 1901, revolutionizing transfusions. His work showed why some transfusions failed due to incompatible blood types clumping together. Want to dive into how he found this or something else about transfusions?
So he's like, what did he do? Why does he is he why did he get a Nobel Prize in 1930?
Landsteiner got the Nobel Prize in 1930 for discovering blood groups, which made safe blood transfusions possible. He identified antigens on red blood cells, explaining why mixing certain blood types caused deadly reactions. Curious about his experiments or another transfusion topic?
Interesting. And how did he do this?
Landsteiner mixed blood samples from different people and noticed some clumped together while others didn't. He identified specific antigens on red blood cells, naming the A, B, and O groups, which explain transfusion compatibility. Want more on his lab work or another angle on transfusions?
I don't get how he did it.
Landsteiner took blood from various people, mixed their serum and red blood cells in a lab, and watched for clumping under a microscope. Clumping meant incompatible types, so he categorized them into A, B, AB, and
I
O.
see. Interesting. And so what's up with this literature like Dracula and all of this? Like, I mean, this kind of rings a bell of blood transfusion. Is it inspired by the medical knowledge at the time, like kind of how it was evolving, what's going on? For instance, Bram Stoker and Dr. Van Helsing.
Yeah, Dracula totally vibes with the blood fascination of the late 1800s. Bram Stoker, writing in 1897, was inspired by emerging medical knowledge about blood and early transfusion experiments, which were still risky and mysterious. Dr. Van Helsing's use of transfusions to save Lucy reflects the era's mix of science and superstition, as transfusions were cutting edge but poorly understood. Want to explore how Stoker wove real science into the vampire myth, or stick with transfusion history?
Yeah, and tell us about the vampire myth more.
The vampire myth, especially in Dracula, tapped into 19th century fears and fascination with blood as a life force. Stoker used early transfusion science to ground the supernatural vampires draining blood, mirrored real medical attempts to restore life via transfusions. Folklore about vampires drinking blood got a modern twist with emerging science, blending dread and curiosity. Want more on how Stoker mixed myth with medicine or another vampire angle?
Sure. Also, talk to us about transfusions like in the First World War, in the Red Cross donation, with voluntary donations in general. Yeah.
During the First World War, blood transfusions became vital for treating wounded soldiers, with Canadian Dr. L. Bruce Robertson pioneering techniques using whole blood from healthy donors. The Red Cross didn't yet have a formal blood donation program, but voluntary donations were critical, often sourced on site from soldiers or medics using sodium citrate to prevent clotting. These early efforts laid the groundwork for the Red Cross's later WEWI blood donor programs, which scaled up voluntary donations massively. Want specifics on WEWI transfusion methods or more on the Red Cross's role?
Um, no, no, it's good. Um, so let's talk a bit about infected blood and how these kind of created problems with blood transfusion, please.
Infected blood became a major issue with transfusions, especially when diseases like hepatitis or HIV contaminated donor blood. Before proper screening, like in the mid 20th century, unscreened transfusions spread infections, causing outbreaks and fatalities. Rigorous testing and donor screening, starting in the 1980s for HIV, drastically reduced risks. Want details on a specific disease or how
Yeah, like
screening evolved?
hemophilia, for instance, or hepatitis.
Hemophilia patients, needing frequent transfusions or clotting factor concentrates, were hit hard by infected blood, especially in the 1970s 80s, when hepatitis C and HIV contaminated supplies. Hepatitis spread widely due to unscreened donors, while HIV infected thousands of hemophiliacs before testing was standard. Improved donor screening and viral inactivation techniques since the 1990s have nearly eliminated these risks. Want more on hemophilia's impact or hepatitis specifics?
Yes, please.
Hepatitis, especially hepatitis C, was a massive problem for hemophiliacs getting blood products in the 1970s 80s. as it was often undetected in donors, causing chronic liver disease in many patients. HIV hit even harder, with contaminated clotting factors infecting roughly half of U.S. hemophiliacs by 1985, leading to thousands of AIDS deaths. Modern screening and synthetic clotting factors have slashed these risks to near zero. Curious about a specific case or how screening tech improved?
Um, no, this sounds good. Um, so so let's talk a bit about the blood groups then.
Blood groups like A, B, AB, and O are determined by antigens on red blood cells, as Carl Landsteiner figured out. Each group can only safely mix with certain others. Mismatching causes dangerous clumping. O negative is the universal donor, while AB positive can receive from anyone. Want to dive into Rh factors or something else about blood groups?
Hmm. So yeah, let's talk a bit about what the antigens are on the surface, what the antibodies are. Let's start understanding these terminologies, I think.
Antigens are proteins or sugars on red blood cell surfaces that define blood types, like A, B, or none for O. Antibodies in your blood's plasma attack foreign antigens. So if you're type A, you have anti-B antibodies that clump with B or AB blood. This antigen-antibody reaction is why matching blood types is critical for safe transfusions. Want to dig deeper into how antibodies work or something else?
Yeah, so the antigens are, wait, which ones are the antibodies, which ones are the antigens, and what's the etymology?
Antigens are molecules on red blood cells, like A, B, or Rh, that identify your blood type. The term comes from antibody generator, since they trigger immune responses. Antibodies are proteins in your plasma that attack foreign antigens, named from antibody for their role fighting invaders. For example, type O blood has no antigens, but anti-A and anti-B antibodies.
I don't get it.
Ma-
Again.
Okay, let's break it down. Antigens are like ID tags on red blood cells, A, B, or none for type O, named from antibody generator because they spark an immune reaction. Antibodies are in your blood's plasma, attacking foreign antigens. Their name comes from antibody, meaning they fight invaders, like anti-A antibodies in type B blood, clumping with type A. Clear now, or
So
want another
we have for blood type AB, we have AB antigen, blood type A antigen, type B antigen, blood type O no antigen. Why no antigen for blood type O?
Exactly. AB has both A and B antigens. A has just A, B has just B, and O has no antigens on its red blood cells. Type O lacks antigens because it doesn't produce those specific surface markers, making it invisible to antibodies, which is why O is the universal donor. Want to know more about why O evolved this way or something else?
Yes, please.
Type O likely evolved as a neutral trait, lacking A or B antigens, possibly offering survival advantages by reducing immune reactions in diverse populations. Its no antigen status means O blood doesn't trigger attacks from anti-A or anti-B
I see.
antibodies.
And what do antigens do anyway? At the end of the day.
Antigens on red blood cells act like molecular IDs, signaling to your immune system whether cells are self or foreign. They trigger antibodies to attack mismatched blood, causing clumping, which is why matching blood types is crucial for safe transfusions.
I don't get it. One more time.
All right. Antigens are like name tags on red blood cells, saying, I'm type A, B, or AB. If foreign antigens enter during a transfusion, your immune system's antibodies spot them and attack, causing dangerous clumping. That's why we match blood types to avoid reactions. Clear now?
So basically, antigen is the passport of the cell, antibody is the policeman, and then if you don't have the country's valid passport, the policeman comes and gives you trouble.
Spot on. Antigens are like a cell's passport, showing its blood type. Antibodies are the cops, checking those passports and attacking if they don't match your body's rules, causing trouble like clumping. Want to explore more blood type quirks or shift gears?
Well, I want to ask you about agglutinins.
Agglutinins are another name for antibodies in your blood's plasma that cause clumping, or agglutination, when they meet foreign antigens. For example, if you're type A, your anti-B agglutinins will clump with B or AB blood, making transfusions tricky. Want more on how agglutinins work in testing or something else?
I don't get what they are. They are antibodies?
Yeah, agglutinins are antibodies, specifically the ones in your plasma that make foreign blood cells clump together. Like, if you're type O, your anti-A and anti-B agglutinins will attack A or B antigens, causing agglutination. Clear now, or want to dig into their role?
So they cause sticking.
Exactly. Agglutinins are antibodies that make foreign blood cells stick together, or clump, when they spot mismatched antigens. This clumping, called agglutination, is what causes transfusion reactions if blood types don't match. Want to talk about how this is used in blood typing or something else?
Well, this is good. Okay. What about ABO antibodies?
ABO antibodies are the specific agglutinins in your plasma that target A, B, or AB antigens. For example, if you're type A, you have anti-B antibodies. Type O has both anti-A and anti-B, which is why it reacts to any non-O blood. These antibodies drive the clumping reactions in mismatched transfusions. Want to explore how they're detected or something else?
So why do they occur naturally due to cross-reactivity with gut bacterial antigens?
ABO antibodies form naturally because your immune system reacts to gut bacteria that have antigens similar to A, B, or AB. From infancy, exposure to these bacteria trains your body to make anti-A or anti-B antibodies against types you don't have, like type O making both. This cross-reactivity preps your immune system for defense but complicates transfusions. Want to dive deeper into this immune training or shift topics?
I don't get it.
Okay, let's simplify. Your gut has bacteria with molecules that look like A or B blood antigens. Your immune system sees these, assumes they're foreign, and makes ABO antibodies against the blood types you don't have, like anti-B if you're type A. Clear now?
Because basically, like, if we have A, we just don't want B to be around.
Yep, exactly.
So why don't we want B to be around? Can't we coexist happily in peace, all of us together?
Haha, love the peace vibe. But here's the deal. If you're type A, your immune system is programmed to see B antigens as invaders. Thanks to those gut bacteria mimicking B, it makes anti-B antibodies to attack, causing clumping if B blood shows up, which can block vessels and mess you up.
But why?
It's all about your immune system playing defense. Your body learns early on to recognize your own antigens, like A if you're type A, as self, Anything else, like B antigens, gets flagged as not me due to similar molecules on gut bacteria, so it makes
But
antibodies to
why
wipe them.
evolutionarily? Why don't we want this mix up to occur? What's the bad thing that would happen?
Evolutionarily, ABO antibodies likely gave a survival edge by priming the immune system against pathogens with antigen like molecules, like bacteria or parasites. Developing antibodies against non self antigens triggered by gut bacteria helped early humans fight infections better, even if it complicates transfusions now. Want to explore how this shaped blood type
Yeah,
distribution?
because I'm
Or
wondering why did we need to attack it if B came about? Huh?
Great question. Early humans faced tons of microbes with molecules resembling A or B antigens. Evolving to produce antibodies against foreign antigens, like B if you're type A, helped fight off infections from those microbes, boosting survival. It wasn't about attacking B blood specifically, it was about being ready for environmental threats, and transfusions weren't a thing back then. Want to dig into why certain blood types became more common in some regions?
I don't get it. I still don't get um why our body feels the need to attack a foreign one, a foreign blood type.
All right, let's break it down. Your body attacks foreign blood types, like B if you're type A, because your immune system is wired to spot anything that's not you as a potential threat. Gut bacteria with B like molecules
Yeah,
train it
why
early.
is it wired like that?
Your immune system is wired to attack foreign stuff to protect you from invaders like bacteria or viruses that could make you sick. Evolution favored this hypervigilance because mistaking a harmful pathogen for safe was riskier than overreacting to something harmless, like foreign blood. So, it flags anything not matching your antigens, like B if you're A, as a threat. Want to explore how this trait stuck around despite transfusion issues?
No, anyway, we'll come back to this. Let's go back to pharmacokinetics. We talked last time, guys. Check out the episodes about pharmacokinetics. Um, but we focused a lot on things like bioavailability and drug administration and all of that. Maybe let's do a small recap because it's an interesting one. Explain to us why bioavailability is oral over IV.
Cool, let's switch gears to pharmacokinetics. Bioavailability is how much of a drug gets into your bloodstream to do its job. Oral drugs have lower bioavailability than IV because they go through digestion and liver metabolism, which breaks down some of the drug before it reaches the blood, while IV delivers 100% directly. Want to recap another pharmacokinetic concept or dive deeper into bioavailability?
No, this is good. Okay, let's start shifting then. Remind us first pass metabolism then.
First pass metabolism is when a drug, taken orally, gets absorbed in the gut and goes straight to the liver via the portal vein. The liver breaks down a chunk of the drug before it hits the bloodstream.
Tell us more, dude. Tell us about the portal vein, the hepatic artery.
All right, let's zoom in. The portal vein carries blood from your gut, where oral drugs are absorbed, straight to the liver, which metabolizes some of the drug, reducing bioavailability. The hepatic artery, on the other hand, supplies the liver with oxygenated blood, but isn't directly involved in this first pass metabolism that oral drugs face. Want to explore how this affects specific drugs or another pharmacokinetic angle?
All right, so yeah, let's go back to the. Um, I'm a bit confused with the first pass metabolism, what exactly it is, Grog, and also, yeah, tell us what the hepatic vein and the portal vein and the hepatic artery do.
No worries, let's clear it up. First pass metabolism is when the liver breaks down some of an oral drug before it hits the bloodstream, reducing how much drug is available to work. The portal vein carries drug-laden blood from the gut to the liver for this processing. The hepatic artery supplies the liver with oxygen-rich blood to support its functions, and the hepatic vein drains the processed blood from the liver into the general circulation. Want me to simplify any part or dive into an example?
Yes, please, I'm a bit confused still. Yes, please, I'm a bit confused still, to be honest.
All right, let's make it super simple. First pass metabolism means the liver eats up some of an oral drug after it's absorbed from your gut, so less drug reaches your body. The portal vein is like a delivery truck, taking the drug from your intestines to the liver. The hepatic artery brings fresh, oxygen-rich blood to keep the liver working, and the hepatic vein is the exit, carrying the filtered blood out of the liver to the rest of your
So the
body.
hepatic vein is the exit and the portal vein is the entry.
Exactly, you got it. The portal vein is the entry, bringing drug-filled blood from the gut into the liver for processing. The hepatic vein is the exit, sending the filtered blood out of the liver to the rest of your body. Want to tie this to how it affects
And
drug bioavailability?
why is the order portal vein, hepatic artery?
The order isn't exactly sequential, they just have different roles. The portal vein comes first in the drug's journey, carrying absorbed oral drugs from the gut to the liver for first pass metabolism. The hepatic artery supplies oxygen-rich blood to the liver simultaneously to support that metabolism, and the hepatic vein drains the processed blood out last to the body. Want to clarify their teamwork
Yes,
or
but
jump to an...
it's a bit, this is sequential, and I'm wondering why this is the order that the things happen.
I hear you, but it's not a strict sequence like steps 1, 2, 3. They work together in a system. The portal vein kicks things off for oral drugs by delivering them from the gut to the liver for first
Yes,
pass
but why did we call it portal vein?
The portal vein is called that because it acts like a gateway, or portal, carrying blood from the digestive organs directly to the liver for processing. The term portal comes from Latin porta, meaning gate, since it's the main entry for nutrients and drugs to be filtered before hitting the
Well, the
rest of
other
the...
two, they are not portals.
Good catch. The hepatic artery and hepatic vein aren't called portals because they don't serve as that specific gateway like the portal vein, which funnels blood from the gut to the liver. Hepatic just refers to the liver, from Greek hepar, meaning liver. The artery delivers oxygen to the liver, and the vein drains processed blood away, without that gate-like role. Want to dig into their names more, or shift back to pharmacokinetics?
Well, to be honest, you haven't given me a good reason why the names are as they are.
Fair enough. Let's dig deeper. The portal vein is named from Latin porta, meaning gate, because it's the dedicated pathway shuttling blood from the gut and spleen to the liver, like a gate for processing. The hepatic artery and vein are named hepatic from Greek hepar, liver, as they simply supply blood to and drain blood from the liver, without that unique gateway function. So no portal label. Clear now? Or want more on
Why
their
no
naming?
portal vein label?
The hepatic artery and vein don't get the portal label because they don't act as a specialized gateway like the portal vein, which uniquely channels blood from digestive organs to the liver for first pass processing. Portal comes from Latin porta, gate, and only the portal vein fits that role, while the hepatic artery just feeds the liver oxygen and the hepatic vein drains it. So they're just named for their liver connection. Want to explore more naming quirks or move on?
Yes, I want to continue exploring this, how their names relate to structure and function, yes.
All right, let's connect the names to their structure and function. The portal vein, named from Latin porta, gate, is a wide, short vessel collecting blood from the gut and spleen. funneling it to the liver for filtering nutrients and drugs, acting like a structural gateway. The hepatic artery, from Greek hepar, liver, is a narrower, high pressure vessel delivering oxygen rich blood to fuel the liver's metabolic work, while the hepatic vein, also hepatic, is a low pressure vessel draining deoxygenated, processed blood from the liver to the heart, structured for exit flow.
So hepatic vein will send deoxygenated blood.
Yeah, exactly. The hepatic vein collects deoxygenated blood from the liver. After it's
Okay,
done
so
processing,
arteries and veins are as usual how they work. It's just the portal vein that's special. And why do we call the portal? And the portal vein is special because it's basically where arteries end and veins start. So there's some oxygenation there still.
you're on the right track. But let's clarify. The portal vein is special and named portal from Latin porta, gate, because it's a unique vein that carries nutrient rich, partially oxygenated blood from the gut to the liver for processing, not a typical artery to vein transition. It's not where arteries end and veins start. It's a vein collecting blood from capillary beds in the digestive system before delivering it to the liver's capillaries, making it a distinct gateway. Arteries, like the hepatic artery, and veins, like the hepatic vein, follow their usual roles. Arteries carry oxygenated blood to the liver. Veins drain deoxygenated blood out. Want to nail down the oxygenation bit or keep exploring the
That's all
portal
right. It's
vein?
all right. Can you explain to us how come this vein is so special that usually because they're usually deoxygenated, the veins?
No worries. Let's dive in. The portal vein is special because, unlike most veins that carry deoxygenated blood to the heart, it carries partially oxygenated, nutrient rich blood from the gut's capillaries to the liver for processing, not to the heart. This unique role as a middleman between two capillary systems, gut and liver, makes it a gate like vein, hence the name portal from Latin porta, and sets it
I don't
apart from.
get it. Explain to us again.
Okay, let's simplify. Usually, veins carry deoxygenated blood to the heart, right? The portal vein is special because it doesn't do that. It's a vein that takes partially oxygenated, nutrient packed blood from the gut and spleen straight to the liver for filtering, acting like a unique checkpoint, which is why it's named portal, meaning gate. Clearer, or want to focus on one part?
I don't get it.
All right, let's break it down even more. Most veins carry low oxygen blood to the heart, but the portal vein is different. It's like a special delivery vein that takes blood full of nutrients and some oxygen from your stomach and intestines to the liver for processing, not the heart. That's why it's
Yes,
called.
but again, why is this vein so special that it has oxygen in it?
Got it. Let's focus on that. The portal vein has some oxygen because it collects blood from the gut's capillaries, where food and oxygen from digested nutrients mix in. Unlike typical veins draining fully deoxygenated blood from tissues, it's special because it acts as a direct pipeline to the liver, delivering this nutrient and oxygen mix for the liver to filter and process before the blood moves on. Want to explore why the liver needs this setup
I
or
don't
something
get
else?
it.
No problem. Let's try again. The portal vein is special because it picks up blood from the gut, where some oxygen from digested food gets absorbed. So it's not fully deoxygenated like most veins. It carries this slightly oxygenated, nutrient rich blood straight to the liver to be cleaned and processed. Unlike regular veins that just go to the heart. Clear now, or want a
No,
different
I don't
angle?
understand why it's clear, and I don't know. I don't, and I don't feel bad about it either.
No need to feel bad. It's tricky stuff. Let's try a super simple angle. The portal vein is like a VIP shuttle, grabbing blood with some oxygen and nutrients from your gut and driving it to the liver for a special checkup, not the heart like other veins. Want to stick with this, maybe with an analogy, or switch to something else for a breather?
I don't understand why. I don't understand why. Why? Yeah, dude. No, I don't want to. No, explain to us why is this vein special and it has oxygen? The rule we learned since kids is that veins no oxygen, arteries yes oxygen.
All right, I hear you. Let's tackle this head on. You're right. Kids learn veins carry no oxygen, arteries carry oxygen. But the portal vein breaks that rule a bit. It's special because it grabs blood from the gut, where some oxygen from food absorption lingers in the capillaries. So it carries partly oxygenated blood to the liver for processing. Unlike typical veins that carry fully deoxygenated blood to the heart, the liver needs this nutrient and oxygen mix to do its job,
And
making the
who and where does the deoxygenated blood? Ah, we have that. Okay. Why do we have a hepatic portal vein and we also have a hepatic artery then? Why do we have two? Why don't we usually have one? Huh? No? Grog?
Good question. The liver gets two blood supplies because it has big jobs. The hepatic portal vein brings nutrient rich, partially oxygenated blood from the gut for the liver to process food and drugs. The hepatic artery brings fully oxygenated blood from the heart to give the liver the energy it needs to do all that work. Most organs get one main blood supply, but the liver's dual role, metabolism and detoxification, needs both the portal vein for nutrients, the artery for oxygen. Want to dive into how they work together or
No,
something
that's fair
else?
enough. That's fair enough. Okay, and let's go back to the antibodies and the transfusion. So, um, how can transfusion of ABO incompatible blood cause intravascular lysis of red cells? And how does this lead to major life-threatening problems, shock, hypotension, etc.?
All right, let's jump back to transfusions. When ABO incompatible blood is transfused, like giving type B to a type A person, the recipient's anti B antibodies attack the donor's B antigens, causing intravascular hemolysis, where red blood cells burst in the bloodstream. This releases hemoglobin and debris, which can clog kidneys, trigger severe immune reactions, and cause shock or hypotension by disrupting blood flow and oxygen delivery, potentially leading to organ failure or death. Want more on the immune cascade or specific complications?
Um, well, I don't understand why this happens.
No worries. Let's break it down. If you give type B blood to someone with type A, their anti B antibodies latch onto the B antigens on the donor's red blood cells, making them clump and burst inside the blood vessels. This hemolysis floods the bloodstream with cell debris and free hemoglobin, which can block kidneys, cause inflammation, and mess with blood pressure, leading to.
Got it, that makes sense. So it's the bursting.
Yep, exactly. The bursting of red blood cells from those antibody antigen clashes is the key. It releases a mess of hemoglobin and debris that screws up your body, causing serious issues like shock or kidney failure. Want to dig into how doctors prevent this or another transfusion topic?
Alright, let me ask you about blood group frequencies. So we have a phenotype of O or a phenotype of A, B, or AB. Explain to me the genotype.
Let's dive into blood group genetics. The ABO phenotypes, O, A, B, AB, come from genotypes based on three alleles, A, B, and O. A and B code for antigens. O codes for none. Here's the breakdown. Phenotype A, genotypes AA or AO, has A antigens, naturally has anti-B antibodies. Phenotype B, genotypes BB or BO, has B antigens, naturally has anti-A antibodies. Phenotype AB, genotype AB, has both A antigens, no anti-A or anti-B antibodies. Phenotype O. Genotype OO. No antigens. Naturally has both anti-A and anti-B antibodies. For type O, the anti-A and anti-B antibodies form naturally due to gut bacteria mimicking A and B antigens, triggering the immune system to make these antibodies against non-self antigens. Want to explore how these frequencies vary globally?
I'm a bit confused, to be honest, with these blood frequencies and phenotypes and genotypes and antibodies. Please explain them a bit more.
No problem. Let's break it down simply. Your ABO blood type, phenotype, is what we call A, B, AB, or O, based on antigens, like ID tags, on your red blood cells. These come from your genotype, which is the pair of alleles you inherit from your parents. A, B, or O. Genotypes and phenotypes. Type A, genotype, AA or AO, shows A antigens, phenotype A. Type B, genotype, BB or BO, shows B antigens, phenotype B. Type AB, genotype AB, shows both A antigens and B antigens, phenotype AB.
Got it. I think I got it.